Okay, hello, Lina.
Hello, Professor.
Hello.
Hello, Lola. Hello, Sarah. Kay, are you lifting hand to say hello or because you want to speak?
I want to speak.
Oh, sure, please do.
Professor, so we need to submit our assignment, right, on Friday. So is it possible to extend the date? Like, I think we all are busy. If
Oh,
today is
sure,
Wednesday,
please, not a problem. It
so
doesn't mean that I'll stop giving you assignments, but you can submit it by Tuesday for everyone. It's
okay,
not a problem.
okay,
And for UK, it will be the easiest because you just repeat basically the things that you already presented.
yeah, I already presented that one, but tomorrow I'm busy
Yeah.
and then I
So
don't think I'm able to do that.
okay,
That's
good.
why.
So next homework is postponed to Tuesday instead of Friday.
Okay, thank you.
Okay. So I will basically talk to you one-to-one. So you will have this board and projector just in case there will be common things, but there should be one-to-one brainstorming sessions today. So I'll try to visit maybe for 10 minutes or longer if needed with everyone. And after everyone is getting happy and confident in the subject of the project, then we can progress towards completion of the projects in more relaxed way, and either at the end of meeting today or next meeting, I can bring in a couple of additional skills, but it will be secondary. After your bright and outstanding presentations yesterday, you all are certified experts in VASP, and you have all tools to successfully complete projects. And even without this course, from now on, you can just do a single author computational chemistry papers. Amount of skills that you have is more than enough for independent career. Okay, so in the previous years, we were I was spending a lot of time in the lecture overviewing projects, but it didn't pay off well. It was no reason to explain project that no one wants to do it. It's more important to do personally. So maybe I will just list everyone and just make a plan of visits. That was huh? That was sorry. So I already have some ideas and pre-made maybe some models for a couple of classifications, but for others we will need to do interviews. So for Boston ideas, yes. Student, yes. Ikeda, yes. For Wina, no. I need to. I remember you were telling ideas before, but I need to refresh in my memory. Maybe you have some models to go forward, but it will take long. For Sarah, very confident, yes. I think it's very good standing. Oh, okay, do not do it, do not do it. It looks like you're jumping from the mountain. I was scared. So, and well, I have some idea, but we need to discuss. And for Boston, there is an ambiguity of models. Maybe there are some, likely yes. For Stephen also, likely yes. Okay, more likely yes. For winner is to be top. For Sarah, it is sure yes. And for Lola, you need to discuss. So. Let's maybe before I start, when I start visiting one to one, it will be boring for the rest. So let's briefly chat. During Friday lecture, I will add general overview of projects, not about with details, not about the individual projects of everyone, but some common grounds, how to organize calculations. And main task for today is to make sure that everyone is happy, has some idea, and go forward. So ideally, it should be something that is interesting for your primary advisor, and you can contribute to your thesis, or something that is easy to do, but and is of interest to you. Size of the model shouldn't be larger than 1,000 atoms. So if you think of something ambitious, it's better to simplify the model compared to ambitious. And there will be details and exceptions, but generally the most constructive way is to explore response of a model to thermalization, whether something will break. It's not. Sometimes there will be some adjustments, but think on this ground. And probably I'll go visit everyone in this list. So if now is 6:10, 6:20, 6:36, 7:10. So if it's very approximate and rough, but if you have some homeworks to do for other classes, you can work on them before the visit, or you can invest time in designing your project if you didn't design, or start computing. Before we go to specifics, there are a couple of things that we didn't include in the labs before, and then the visitors and readers of your reports will ask about. So I'm always forgetting, but please remind me or explore yourself. What should one do to repeat a calculation that you are doing on VASP with different function, specifically this hybrid function, which is more precise? And at least, so if one looks into the bin card, there should be directory that ends with this letter and with this code, and it will be a template in card that will give higher precision. So I'll probably start visiting Boston and Steven. So I apologize if you do not have any entertainment at this time, but I need to go over. Are there any emergent questions?
I just want to make sure. So for each student, you want to discuss about the project with them?
Sorry, I was reading. Just a second.
Okay.
Lola is asking, finishing before 8 p.m. Oh yes, sure. I need to report myself to return home before 8 as well. So today we should be quicker than before. Okay, sorry for being in, yes. Oh, okay. So Boston wants to sacrifice his priority. I selected him because I saw him personally. Okay, let's. So then you're last. The last. Okay. 7, 10, 6, 26, 30. So Lena, I apologize. I'm not very much focused, but please repeat your question once again.
No problem. So I was asking, so will we discuss about the project individually? That what's the plan?
Yes, it is the plan. And we can continue emails, we can visit personally by Zoom or Skype, but time goes by really quick. And since now we have a designated time for the class, we better do this interview right now. And
Yeah, okay. Yeah.
so the goal is that by the end, like in 1 hour and 45 minutes, everyone will have a molecular model for the project.
Yeah,
So,
yeah.
if you already have an idea what you're going to do, you can start either downloading it CIF file from database or drawing it in Avogadro or Gauss view so that you speak specifically.
Yeah, that's perfect.
Okay.
Mm-hmm.
So.
I'm thinking to come to the classroom.
Oh yes.
Because it will be maybe better to discuss in person.
yes, it will be much easier.
Maybe I will leave and I'm coming to the classroom now.
Okay, yeah.
Okay, thanks, Dr. Kelly. Thanks.
See you soon. Okay, so I will be chatting to Steven right now, but if there are some emergent questions before I go into the sequence, just speak out loud. We are on the sound system, so I will be in the class. And we can focus. Yeah. But the sound system goes there. Okay, yeah. So after Steven, next will be Kea, and then Willie will come in person. Yeah, I have an idea. Well, I have an idea for your project, but you can think what you want, and we'll discuss. Whew. Okay. So it's still it is done. Now, okay. Okay, would you like to? Yeah.
Now it's my turn.
Yes.
Okay.
Let's chat first, and then I'll share some images.
Okay.
There could be sound feedback, but I will try to remove it immediately. Just because there are some materials on the other. Microphone. Okay. So no sound feedback. Whew. So we were chatting yesterday that there are ideas of your advisor, Professor Shah, of exploring the twisted conjugated polymers, right?
Yeah.
So this is quite a simple thing that we can practice now, or you can practice alone and then share models with me before running. Let me draw something on the board, and then if needed, maybe draw it together. So I will try to use thick lines and large because camera is far enough. So suppose we have this polyacetylene, right? And now I will just draw it once again with less visualization. So, and I skip hydrogens, I put on the carbons. So, the energy of homologous, if you put like, 3 hydrogens terminal group, which is CH3, the homologous will depend on the length of this oligomer. If it is a final length.
Sorry, Professor. Can you speak loudly because you were marked? I am not able to hear you properly.
Do you hear from this microphone, or maybe it goes from the different one? Yes. Yes.
And I think now it's okay.
Okay. The HOMO-LUMO gap, which is a key feature of conjugated polymer, depends on its length. So in last 3-4 labs, we were dealing with infinite lengths, periodic boundary conditions. But in reality, there are oligomers and there are polymer chains of the certain lengths. The length does control HOMO-LUMO gap. So if you take the Gauss view tool to change the hydral angles, you can select a bond, rotate the hydral angle here so that this stuff will be oriented not here, but for example in the different direction. Right? And after such rotation, the efficient length of the polymer will be shorter.
Yep.
So if you create several samples by making it shorter, then like arrange it L1, L2, L3, L4, then one can just make a single point calculation and find HOMO-LUMO gap for each of them. So basically, it is what your advisor suggests. So it is some very quick calculation that will make him happy, and it is. I think it is not enough for this class. We can do something more more advanced, but you should do it first to get credit from your group. There is another thing that I want to suggest, and you are free to accept or reject, but it seems very, how to say, promising.
Okay.
This thing standalone can be on seems as a paragraph in a paper, and the stuff that I'm going to suggest could be like standalone calculation. Oh. So if you explore open sources, do not even not specialized literature, just Wikipedia on conductive conjugated polymers and polyacetylene, you'll see there is a section on doping. If you doping, I do not hear you.
Oh, okay. After conjugated
Do not
polymer,
hear.
can you hear me?
Try speaking.
Are you able to hear me right now? Is it okay?
Try speaking once again.
Uh,
Oh,
Professor,
okay,
it's
better,
okay?
better.
Okay. So after conjugated polymer, you said something. Can you repeat that one again?
The word doping,
Before doping.
open sources, just Wikipedia on conjugated polymers and polyacetylene. If you just read open sources, you will see there is a paragraph telling that one of the key experimental way to control their properties is to introduce doping.
Okay. Doping.
And doping, which means impurity, something foreign for this material that changes its structure. Most typically, electron donating or withdrawing agent.
Okay.
And if one has a bulk of polymers, like when several threads are smashed together and there are regions with positive and negative doping, this can be a source either for light emitting or for photovoltaics. So if you try to model such material, it will be not just academic interest, but practical application.
So that one is possible for the same model, right?
Yeah. Do you have the model on the link? Can you download and share it on the screen, or you want me to do it?
So you just share with me today something like.
Yeah. Can you download and open it on your screen in graphical format, like in VMD or in yeah, VMD would be the best.
Professor, so that's the link. How to download that one? I don't know it.
It is a directory on the NERSC.
Okay, I need to log in there.
Of course.
Okay.
You need to log into NERSC every morning.
Professor, can you do it now? Because it's I need to it's because I'm in the Mac system, so there I don't have. I need to okay. I need to go to the. Yep. Um, let me. I need your permission for that. Oh. Yep. Professor, I need to connect my laptop with my office desktop. Just.
Yeah, please do.
Yeah, I already logged in that one.
Yeah, it's better if you do it and share. I'll try to
Okay, okay.
back up, but
I'm doing it. I already logged in that one. A few steps needed. Just. Mm, mm, okay. Professor,
yes,
um,
I can stop sharing if you already downloaded it.
uh, I don't know how to
Oh,
share.
I need to make you co-host. Just a
Yes.
second. Uh, host. Yes.
Yep.
If you load more
Mm.
ones.
Are you able to hear me right now?
Yes, I can.
Okay. So I use the link you give me here,
Yes.
and after that, how to download that one, Professor? I don't know.
So R sync
R
RSYNC yeah, minus
A B
AV like
A
Alpha Victor AV space then user name no not like username KILIN photon yeah add photon
phone, then the previous name, right?
yes
Edu,
yes
right? Then enter.
yeah no column 2 dots column
Okay. Is it okay?
yeah and now path from where you are logged on the photon copy paste here the path where you assign for the project on a photon.
In a photon, I need to make a directory also.
You should have a directory for the project. Are you logged into photon on us in any other tabs?
Yes. Let me do-
Yep, somewhere.
Oh, what is this?
Okay, project is good.
Oh, it's already there.
Click, double click.
Just one minute. Yep, this one. And oops. Okay.
Codopt 2.
It's already there.
No. Copy the path. Copy the path.
Proceed here and
Yeah, paste
okay.
here. Uh, just a second. Make arrow left several times. Arrow left.
Arrow left.
Yeah, into the whole way between minus AV and name of the host.
Okay. Yes, there.
Yeah, put star space star space
Space, star,
star
and
space.
there is space off.
And now enter.
Okay. sending this file to that one, right?
Okay, yeah, and now try to look for update.
Yes, yes.
Maybe download Concar or download or maybe Outcar. Download and open with DMD.
Concat or outcut?
Doesn't matter.
Okay, concat. Okay, let me download it. Where are you downloading? Just 1 minute. No, I need to download it again. Where is it? Okay, let me download this one. Downloading. Downloading. Document. Okay. Outcut. Load. Command. Load. Oh. Oh. Right.
Okay, just let's quickly look on the model. You do
I
some
have
visual.
to do the rest of the part. Like.
Can you
load
rotate
by-
it a little?
Yeah.
Rotate it so that it will be perpendicular to like oriented in the plane. More, yeah. Keep rotating, yeah, about this way. So what do you see?
Oh. Why there is a gap?
There is a empty. Yes, you see this phosphorus fluoride, and can you navigate higher up and right a little, up and right? Yeah, stop here, and
Yes.
here is the sodium. So this sodium and phosphorus fluoride are PNN doping, which sodium donates electron and phosphorus fluoride withdraws electrons. And due to presence of these things, there will be, upon photoexcitation of this middle strands, the charge will transfer to either one or another side.
Mhm.
So just try to optimize and characterize this model as we typically do.
Okay. Okay.
You can do something else, but I recommend, and this should be very interesting, and you will not regret.
Yeah, I will not, because last time you changed my presentation topic, and that one is much better than the previous one.
Okay. So I think we are done. We will keep talking as we go through semester. You can stop sharing. And
Yeah.
I want to give a brief comment to everyone. In the previous labs, we, in both Gaussian and VASP sections, we were not spending much time on the geometry optimization, which I was doing it intentionally so that we do not waste, you do not waste time on this procedure. But if you do productive results, optimization, if you do something for with scientific interest rather than trained skill, optimization is challenging task that takes more time. So I just attract your attention to this problem, and as we go through the rest of the semester, several meetings, I will focus on it. So in order to get optimized geometry, it's not sufficient to request like 100 steps of optimization and wait until they're done. If Wasp doesn't stop automatically before it hits the end, your threshold of steps, it means your structure is not optimized. And then it means that one needs to take cont carve from initial optimization, rename it as a postcard for next step of optimization, and run it again. If we would have as nice system administrators as Steven in all supercomputers without wall time, we can just request 1,000 steps of optimization, it should be enough. But Photon is not as strong to upkeep all our needs, and productive clusters outside, they typically have wall time for like 24 hours, and big systems, for most systems, it's not sufficient to optimization. So anyone needs to take whatever was done on one step, then go forward. Another trick that transition from different stages of optimization is arranged in the same way as Kay was teaching us yesterday, as transition from heating to molecular dynamics. When we are going from heating to molecular dynamics, we inherit cont carve and rename it into postcard and then continue. Cont carve means continue. And originally it was designed for optimization, not for molecular dynamics. So just try, we will keep interviews shortly, but try to make effort with optimization, because if system is under optimized, results are not trustable. So for learning purposes, it doesn't matter, but for scientific research, it's a key portion. And even a little change in geometry may result in drastic change in the progress. Like in molecules, change of the bond length by 1% like may give like half electron volt or electron volt in energies. So it's a serious thing, just giving you a little warning. And you were sending feedbacks and so to say complaints that several, like me and my classmate, are sitting by the terminals near each other, doing the same problem, getting different answers. It happens because we didn't pay attention to optimization. If one optimizes to match the threshold of precision, then the results should be the same. Just before, we were not paying attention to it because it would delay the rest of the process. Okay, done for this little announcement. Okay, do you have quick questions? On your subject, or we are done? I don't hear you. Your microphone is muted.
Oh, shit. Oh, now you can hear me, right?
Yes.
Professor, is it possible to talk with you later?
Oh, yeah, sure.
Yeah.
Yep. But for your like for our formal interview, we are done. You have an idea what to do for the project. Okay. Yeah. Thank you, Kay.
Because I need to think about that, what you said. Then I have some question.
Okay.
Thoughts,
Yes.
those
Yes.
kind of things. That's it.
Yes. Yes.
Okay. Thank you.
Okay. Now Winnie is next. Again, while I'm talking to Winnie, if anyone needs to speak, just speak out loud. We are on the same sound system, but I will join.
was thinking to have models like as we did at model heating for the nuclear dynamic. So if we apply temperature, if we increase the temperature, we can just slab that that we can get the situation. So first it's connected to each other and then
What is the model? So you're talking about procedure, but which objects are you interested
in my project, I'm studying the use of thermal stresses. One for the living.
in? You need to teach me what they are.
Yeah, okay. So what I did, I applied this, I attached this ligand to AFM.
Mhm.
And I applied this step close to here, that can then inside the cell, and when I just hold it like up, it's like applying a force, and then when this goes in contact with here and these two molecules
Okay.
attach to each other, so when it's go back, it will unbind.
Mhm. Very good.
So
Now,
I think
what are they chemically?
okay, so this is the process that they are composed. This has a part of lysine.
Okay.
And
Yes. Very good.
And this has complete structure. It has binding site. It can bind easily to caffeine.
Mhm.
I'm not sure about it. I can use the name of it, but it's complex.
But we need atomic structure
Yes.
of that.
we need the other structure.
And they
I
can go.
was looking at this paper.
So
They
I,
did some
and I will,
for the same.
my IQ will be not sufficient to understand. So you need
Yeah,
to
yeah,
help.
yeah. It's so because I'm just with this because of measuring the force, and
Mhm.
then I'm interested to get information about binding and
Okay.
like K on place, for example,
Yes.
K on place.
Yes.
This is what important.
So that means tell something about you.
Yeah.
Just okay. So we need to find online the PDB in the PDB. And then just download and bring it in all of that. If something goes wrong with this, I see the word metal here. What one can, I don't know why it is, but instead of this like key lock system, one can
Yeah.
take this stuff and just look for binding energy to pure metal. This will be a very quick and simple calculation that will move to the center of the value.
Yeah, we could replace this again with like
Can you replace this one with metal? Because this one to begin is very, yeah. This is very simple and small.
Yeah,
molecule,
yeah.
I know where to take. Yes. But for your agent here, is like maybe I will talk to someone else, but you need to download the PDB and open it in Avogadro on your screen. Here,
down.
What?
This is not.
no, next. This 8.
Here, it's what
Oh, then you can, are you,
I just use Go.
you have those on this computer or on this phone?
No, I just using Go. I didn't download it.
This is fine. But you will be more productive. But something installed in this
So can
computer. Are you?
download it?
Oh, yes, it's good. If you have password, if you have like system manager, if you have system password for the computer, like are you, is it only your computer, no one,
Yeah, it's on.
so when you install software, you can type password or just put your finger for define yourself. Okay, then just look online for Avogadro, download it. Real quick. If you will be just upload stuff to photo and visualize things with but it will be delaying your productivity. You just
So for this project, we have to look at the, yeah.
not anything like that.
Yeah, I
And-
haven't used it.
But I'm suggesting the optimism. You will not regret. You will optimize your efforts and you will do things. You will do that. Feel free to chat to other Western years. I think like Julia and Sarah, they have avocado they use so they know that avocado doesn't have. So, so we can do it right now. Just take avocado software because otherwise it will show the fourth of yeah. Open on trade. Um, let me concentrate.
But how, I mean,
So it brought you to
is it
this.
a trusted?
Huh?
Trusted zone or?
Sure.
Yeah.
So I'll go away, find this agent or it creates biological systems. It's PDB.
So is it,
Type the name.
okay, yes, PTB now?
Type the name of this agent. We'll cut it from the paper.
Okay.
PDB. Should be.
Yeah.
See users.
Was, something.
If it fails, we can just go with. Yeah.
Where's the button? Okay.
This is beautiful of the animation. It's going to bind you. So,
So the things that we know, that is the binding side, especially the
Yes.
binding side, can bind to captains. Not with the magnetic item or magnetic item.
so.
Yeah.
And what we paid
will do it.
Yeah.
So are there any ways to small?
Yeah, yeah, I can find the
So-
small one. I think
Please try to and try to do it as a priority because we or at latest more we need to have the model.
Okay, so we have this and I need to pass it.
Yes.
I will-
And here it looks like it will be more than double that. Oh, okay.
If I find just the molecule or the things that might specifically do this,
Mhm.
we can just use this part without.
Yes, yes.
Okay. Okay, so I will do that.
And please send me description. Concourse or foundation of this. I do not probably much. So we before then already it's every.
Yeah, so I will just confirm 100%. So I have this part and I need to find the other
Yes.
part, the content of this. And then I will go to
Mhm.
the model while there are.
Yes, and how to attach them, I will see, but there is some, at least model of this acceptor site, when you say.
Yeah, yeah, yeah. But I'm talking about the procedure.
Oh.
That's something we cannot know.
Let's discuss procedure after this step,
Okay.
because it is a bottleneck, we will be.
Right.
And if you complete it, clean, you will not be.
Yeah, I was asked about that.
If you do not fix it, let's say within 24 hours,
Okay.
it will be, will be to just look for social energy, Zoom to that. I don't know if it will be useful. But I can write it quite easy with both sides.
Yeah, I know, but I was thinking, as this can be approved within.
I understand. But with this one, then,
Yes.
since you don't have this, you will do it at least something.
Yeah, I like that. Okay. Yeah.
It
It
may-
will be at least like in the future, I can.
Mhm.
It's not, it will be useful.
Mhm. Yeah. Like.
But I will do my best
Please,
to find
yes, and this can be also if you have free time and energy.
PMP.
B and D.
Yeah, I have.
Oh, okay. Okay, let's try to open this.
Yeah.
Just
Okay, can I search?
Oh, yes, yes, yes. Okay. Oh, my pleasure, my pleasure. So done this, now I've got ideas that we still work on models, but with some progress. So Sarah will be next, but I see Kia raised the hand. Kia, would you like to tell?
Yeah, so Professor, like, could you please check your email?
Okay.
Yes.
Or probably you use different one. Oh. Sure, sure. Okay, the answer is positive.
Okay. Yeah, because I'm.
Yes, yes, yes. I understand the circumstances. Not a problem. We do not need to discuss in public, but
Okay.
I'm fine.
Okay, Professor.
Okay, so next is Sarah. So let's chat first, and then go to look onto on the files. So you are looking for adsorption of amino acids on clay, right? And I saw the answers that you typed. So we will discuss example practical samples that may work, but if something is missing there, you may redo building of the model from databases. And please correct me if I'm wrong. I was just reading from open public sources that clay is like mix of silicon and aluminum oxides, and some of the aluminum or silicon can be replaced by other metals like iron or whatever, right?
This one.
And they are, instead of being three-dimensional periodic bulk, they are like two-dimensional sheets. Okay. So do you maybe if you share a screen will be more productive and
Oh, I'm not able to share the screen.
oh yes, sorry. Host.
So this is.
So let's speak about this thing a little later. You can type the TOP to prevent it from logging off. Type TOP. Yeah, enter. And now let's go to a browser, just web browser. You are sharing only one window. If you would share the whole desktop, of course, if there are no secrets, we can discuss several things.
So what should I do now?
Just bring up a browser.
You mean the Chrome?
Yeah, and open new window.
Oh, sorry, I can't. I don't know what's the name of this.
Or new tab,
Okay.
and just type something like amino acids. Amino acids, and select images like under amino acid. Yeah, and basically anything will work. Let's scroll and find the. Oh, okay. Let's click on the one that at the top line at the rightmost.
This one?
Yeah, yeah. Just scroll. Okay. Okay. Yeah. So this classification is a little simpler, but in other classifications they use 5 classes. So can you scroll up for showing polar and non-polar as well? Yeah, non-polar. One can start with the simplest one with glycine, but if we do something more advanced, we can easily do simpler. But you know better what is your thesis goal, what your research goal. But maybe you are thinking about systematically scanning over absorption energy for all of the natural amino acids. But as a little simpler task, one can take one of each class because they have some similarities, and to make it even simple, one can just take one. So also, if you see for positive charge, in green box, this lysine, can you focus on it with mouse? Yes, it has this NH3 plus group,
Yes.
right? No. This one on the top, yeah, on the bottom.
Amine. This one is amine group, yeah?
Yes, this amine group. And can you focus your mouse on it? No, one of the top. Yeah, this one, and then move mouse to the right. Slowly, more to the right. Carboxylic group. So carboxylic and amine group, they will bind to a next amino acid and form a polymer. And if instead of one amino acid you have some peptide, some protein, then those groups will be not available for binding. So it's only if long polymer starts at the surface. But it was the question that we discussed in the email. So if there are some reasons, one can try to orient this carboxylic or topside NH3 plus group to the surface, but it's highly unlikely that this binding will happen. Mostly, it seems reasonable to use this bottom group, and since the clay surface is enriched with oxygens, which are electron withdrawing. It hypothetically, again, it's not my project, it's your project. You probably read more about it. But hypothetically, the oxygen-rich oxide surface will have affinity to attract positive groups. So this plus charge groups will be highly likely absorbed. The absorption energy will be bigger. So this lysine or arginine seems to have larger binding energy, and maybe we can do it as a next little calculation. Let's scroll down. So negative charge, I'm not sure, but intuitively it will not bind to the surface. And polar, your mouse is, yeah, tyrosine. It seems interesting because the aryl, this aromatic ring, is well defined, and even if we mess up with optimization, this aryl ring is hard to damage, so it will resist to human errors, and it will be a good reference point on one hand. On another hand, it's rather big molecules, so that we create simulation cell with enough space, and later on we can just modify the side group and explore others. So if your time is and effort are limited, maybe starting with this tyrosine could be just quick thing to start with. Okay, now please bring up the NERSC window and do Control-C to stop this. Yes. So can you do CD space period? Enter. ls minus OT. So there are several examples of different classes, but they are underdeveloped. Their amine and carboxylic groups are missing, like oxygen or hydrogen. In these directories there are amino acid parts without full carboxylic and full amino group. They are just taken from infinite polymer, and if you want to pursue them, you need to manually add groups. Now let's go to tyrosine residue 1 where you originally were. TY underscore. Please type, yeah. Okay. So type grep space n and with uppercase N up down, N up down, N up enough space space out car. Okay, so we didn't fix it. Now type more OZ-car, uppercase OS-Z-car, OS-Z, OS-Z-I, there is letter I, OZ-car, I, car, enter. So you see, you can just scroll and until the end. So do you see the last record here? Last line, yes. So what does it tell? MAG equals 5.
This one?
Uh-huh.
Oh, I forgot what's like this. It's
So let's. I will not answer. I will just prompt. If you please type tail space minus n 2000, about maybe 1000 space out car, vertical line, vertical line, vertical line.
oh
No, up, arrow up, vertical line more.
More?
Enter. Space, hit space, hit space, hit, scroll up. So look when the occupied converts into unoccupied. Maybe scroll up for the first spin component. Yeah. Stop. So 198, right? Now scroll down. Slowly scroll down. Hit space. Huh? So, and here is 193.
193, yes.
Right? So 198 and 193. So what will be 198 minus 193?
5.
Yes. So it is a number of alpha electrons. 5 is bigger than beta electron by 5.
Yes.
So it means this system naturally wants to have this spin polarization. So the calculations for this clay material without I spin equals 2, without spin polarization, is predominantly wrong. And what you are looking now, calculation with I spin equals 2 is correct. So did you download the structure to your local computer?
No.
Please, well, you can open it in. Do you have Photon?
Yeah.
Direct type TOP here. Just prevent it from. Yeah, enter. Go to Photon.
I have Photon in another tab.
Yes, excellent. Type CD. Just go one directory up. Period. Enter. LS. Oh no! Let's go to your final project. It will be more healthy and yeah. Go to final project. Type PWD. Copy this path. Now go to directory with Cori. Control C. Stop this TOP and type rsync, RSYNC from the word synchronization. Space minus AV. Star. It looks like we are in NASA. We are typing space, star, space. And now user at server, like Kevin at Photon. Yeah. At Photon.
Moodle.edu, right?
Yes, now two dots, colon, no space, right? Colon, and now paste the path.
It needs one space after column or no?
No space, paste the path.
Okay.
Yes, now slash, add slash, and hit enter. Type the password. And now let's go to the Photon directory. Oh, type TOP here in just so that prevent it from enter. Go to the Photon directory.
Here.
Yes, and try to download maybe outcar or concar to your local computer and open it in BMD. Yeah, double click.
Still.
There is a green circle on the. Oh, if it doesn't, yeah, green circle to reconnect. Reconnect, yeah. And now try once again, double click. Scroll up, double click.
So
Just concar or outcar, just one file. Download.
this one is okay.
Yeah, it's good. Download to local computer and open this indeed. Postcar. Concar is the same as postcard. Yeah. What? And also, can you go to representations once again? Representations, periodic. Can you click periodic? Periodic. Slowly move. Yes. Make plus X, minus X. Click. Maybe plus Y, minus Y. Yeah, enough. Now let's go. And in the main, yeah, you can hide this one. And in the main window, can you go to display? Display and select orthographic. It's a third line instead of perspective. I know that some people like this perspective, but I'm getting dizzy and headache of watching this. Okay, now just look around.
Excuse me, what type of clay it is? It is kaolinite, not kaolinite?
You should know better. I even don't remember where I took it, but yellow is silicon, pink is aluminum, and the cyan is iron.
Yes. I am not sure about type of clay because most of them are the same kind of atoms. So they have, but they have different names and properties.
So you need to teach me and the whole class about clays. I saw today your poster on EPSCoR meeting as a first author on clay. So you are a certified expert on them. So you should know better what it is.
Okay.
So the simplest way, like if time is really limited and you want to complete your project in half an hour, you optimize this model fully, make sure it is fully optimized, and then you do the following trick. Open the same model maybe in Avogadro or GMO, because VMD doesn't have the editing tools, and then out of this model create two additional models. So first you cut everything except the amino acid, but keep the same size of the cell. Second, you remove amino acid, keep clay and the water, and keep the size of the cell. Then you have total 3 models: hybrid, clay with water only, and amino acid only. So you find their total energy and subtract them from each other, and this will be binding energy. I think you are doing the same with molecular mechanics.
Okay. So,
So
how
yeah, please. I think you may have some questions right now.
yeah, of course. How to calculate the binding energy?
I already told you. It is what I was telling you in the previous sentence. Let me repeat.
So what about the direction of the amino acid?
Direction of what?
The amino acid. For example,
What is?
uh,
Oh, amino acid.
yeah, amino acid. For example, it should has the interface with the amine side, or with the carboxyl side, or with the side chain.
No. When we looked on the Wikipedia site, I was trying to give an argument that we need to look only on the side chain interfacing clay. Because this amino acid and carboxylic, they will be disabled from binding in real life, because they will be already bound to the whole peptide.
Okay. So just the side,
Huh?
just the side chain
Right.
should have.
Right. Yes. Yes. And I am not insisting. You are already expert or growing expert in this area, but I'm suggesting the shortest path so that you do minimal effort and get maximal output in a short time. So later on, after the class finishes, you can continue and do more systematic study. But if time is up, this side group towards clay seems more reasonable. And for comparison, maybe one can look on another amino acid which has the positive chain, lysine, right?
Yes.
But maybe this one to start with. So your question about amino acid, I'll try to draw here with big letters so that you can see. So A plus B is a full model that you have on the screen. A amino acid, B clay plus water. So for each, you have this one, right? And to generate A and B, you just remove part of the cell. Just do it. And then for each of the models, you compute total energy. Total energy A plus B, total energy A, total energy B, A and B. And then binding energy is defined. So it's not a fantasy of us here in class, it is exact definition. So you take total energy of A plus B minus total energy of A, okay, minus total energy of B.
Mhm.
So you do 3 calculations, 3 optimized geometries, subtract total energies, and in some communities, it's already a publishable result because it can be compared with experiment.
Yeah. Good.
Okay,
Okay, thank you.
make sense?
Yes.
So
So-
there, just a second, there is an additional thing that maybe is not expected in the community of people who deposit amino acids to clays, but here for this class and for curiosity, it's interesting. If you practice these stuff that we were doing in the labs and that you were presenting yesterday, if you draw, if you build the spin polarized dose, you were showing it for triplet, but here it is higher multiplicity. It will be an interesting thing. And one can also redo calculations with different n up down. Right now, it is equivalent to 5. You can check if n up down equals 3 and 1. I expect that for any other n up down different from 5, the energy will be drastically bigger. And the amino acid may stop binding to clay. One thing. Another thing, if you take this, if you plot spin polarized orbitals in the same way as you presented yesterday,
Mhm.
you will find something really beautiful. It could be maybe not directly applicable to main goal, but it will melt the hearts of the reviewers when you go to publishing. So because you have iron here and spin polarization, the HOMO-LUMO orbitals will be localized on the iron.
Mhm.
And it will be like several of them. So like HOMO, LUMO, HOMO plus 1, LUMO plus 1, there will be 5 spin up and 5 spin down orbitals on the iron because it has this d orbitals are inside the gap, and they are affected by nearby oxygen. They determine how they shift up or down in the density of states. Okay, any questions?
Yes, so I should create 2 more models, one for just only clay and water and one for the amino acid, right?
Yes.
JMOL.
Yeah, well, JMOL will be quicker. Of course, some people can erase atoms just in a command line, but I think it is too crazy. If you decide to do it, you just look. So all atoms with coordinates with z projection smaller than certain value can be erased, right? And it will erase all those. But better do it in JMOL. It's more intuitive, and you know how to check it.
Oh, okay, okay, gotcha. Thank you so much.
So you are done with your project?
I will do my best, and if I have question, I will reach you out. Thank you.
Okay, yeah, thank you.
Thank you so much.
Kay, you raised hand again?
Yes,
Or,
Professor, could you please allow me to leave right now because it's
oh, sure,
around
yes, please do.
7:46.
Please do, please do. And connect whenever you need, if you have
Yeah,
questions.
and okay. Thank you so much, Professor.
Yeah, no
Okay.
problem. So Sarah, yes, done here, and then we have Lola, and then Boston. So I will be inside the classroom, but if anyone needs to talk, we are on the same sound system. Sorry.
No.
Oh, yes, absolutely, absolutely. Sorry that our meeting takes so long in the regular non-paid time. Maybe we should arrange one. Okay. Yes. Okay, yeah. Okay, thank you. Okay, that's fine. Oh, finally. So I'm not sure. Click on this carbon. Oh. Just try it because girls in the поставили использование double. Oh, yeah, yes, yes. But okay, very good. So, madam, we need to let people. Okay, thank you.
Sarah, I see you are online. I'm going to talk to Boston now. Let me know if you have some quick questions or you want to speak after we are done with Boston.
Yeah, sure. Professor, for deleting the part of the model, as you said, for JMOL, I should open the postcard file into the JMOL or another file?
I've understood your question and I do have the answer. I'm just arranging words into sentences. So, if you have new version of JMOL, like version 13 or version 14, they allow erasing atoms directly. They allow editing. The latest version of JMOL allows editing postcard, and it allows to read postcard directly. If you have older version, or if you want to be a little independent from from the mercy of JMOL, you can convert postcard into XYZ, and then open it in Avogadro and edit, or in old JMOL and edit there. But yes, it is an important skill that I may have missed. Kea was briefly mentioning it, but I don't remember if everyone placed attention. So after you do either optimization or molecular dynamics, there is a command that sounds like x dot to XYZ. If you type it, it creates a file named movie.xyz that contains snapshots of the molecular dynamics. We can maybe I can do a quick demonstration. Well, let me talk. Let me ask Boston how patient he is. Are you okay if I show something to Sarah and then go to you, or you are already tired to wait?
That's fine.
Oh, thank you. Thank you. I appreciate it much. So, Sarah, I'll do a quick demonstration. Maybe you. So I will. So suppose I already have on my local computer Concar, or I want to be specific. Let me be specific. So this command is available both on NERSC and on Photon. I'm just a little tired to log in over to the servers. Right now it will give error because I'm dealing on a computer. I don't have X.far, but when you download, when you have all formats of VASP in the directory, this command creates file named movie.xyz, and I have it already here. So it converts the outcar into XYZ, and it has, you see, there are several snapshots. They start with again, over and over again, number of atoms, force, and then it goes list of atoms updated. So since I need only one snapshot, I can do minus n, then I put number of atoms plus 2. So if number of atoms 108, I do 110. Move the Z into test. Now, what do we see? So it has 110 atoms, more yes. It's just one snapshot of the structure, and then I can open it either in JMOL. And then go forward in erasing atoms, but by doing this little click on X, right?
Yeah. So just manually, I should delete this.
Yeah, just erase.
And then again, I should save another XYZ file from JMOL, yes?
Exactly. Save, open new directory, both on your computer and on servers, and convert it from XYZ to POSCAR in the standard way as we did before.
Yeah,
Oh, I need to be home at this time. No, we'll talk. Sarah, do you have more questions?
For the binding energy, excuse me, the last question. You said that I should calculate the total energy for the whole system at first. So for this total energy, you mean it is the Fermi energy that we had in the outcore file?
No.
Was different, yeah.
In the OSI car, the next number after F equals. So this is the total energy.
Oh, I see.
And typically it's bigger, it's negative large number. So let me, maybe I have your model open somewhere. And this is here. So for your system, it will be minus 653 electron volts for combined system. And without amino acid, it will be maybe minus 600, and the amino acid itself will be about minus 50 electron volts. So the binding energy should be of the order of like electron volt or half electron volt, something like that.
Okay.
Make
thank
sense?
you.
Any other questions?
No, clear. Thank you.
Okay, yeah, thank you.
Oh, can I leave the class?
Oh, yes, absolutely, absolutely.
Thank you so much. Have a good night.
Thanks, Steve. And probably we can stop recording since, oh no, KMA joined. Whew. I don't know, whatever you want.
I don't know where. Just take June to run. I read a little bit of that. Yeah, I can't say I'm I did, I read through, did they talk, look like they're talking about like graphing. It's, So the Dirac cones, I'm not very familiar with what those are, but I know that they are that, but I don't know what those are. In a sentence. Where you find this? So, first of all, one thing. Okay, because I'm still having problems with it, so yeah, I'm gonna put that in class or it's gonna put on tonight and send it. Yeah, I think, do whatever you like. And just, that's-
Yeah, so just business colorize and like characterize and like on those spectrum, I think the battery you're seeing.
Right. And then we can thermalize.
Basically everything you've done in class.
Exactly.
Applied to the business colorize.
Yes, there is under what is called the draft. It has to be pointed out for the language. It appears to them. See, and this part. Even something that also.
Well,
Some people like challenges.
I was gonna say, even if I do find it to be really difficult, I still really like to learn how to do it, because if I don't, I'm not actual research. So that's something that looks it's very beneficial for me to learn. Because like I said, right now I'm struggling with them. So even just keeping calendar optimized very well. Just out of it, because I thought I built it pretty well.
Yeah, share with you, I did for the same. But,
Yeah, definitely true. I haven't watched TV in months. The one that we have.
Although there are some. Results, but oh. Slash means the root, there are.
Oh, my bad.
Check.
You want me to just do a brand new run, so.
Yeah,
No.
just, yeah, happy. For. Start with each.
cast. So when the job starts. initially remotely, when you set it up. Likely your memory is probably. I don't remember off the top of my head. Two Ds, you mentioned about 2 days. It should be done. I just. Is this days, minutes? Oh, just days, hours, minutes, or is it hours, minutes? Hours. It's now. I won't keep you here. Looks like you're already like this.
I was getting criticism from close family member asking like, are there really students after 9 p.m.?
Yeah. Thanks for staying so late.